All right, good morning, everybody. We're ready to start our next panel discussion. This is going to be a great one that we have done in years past and people really enjoy very much. I want to introduce our panel, and they're going to do a little introductions themselves. You are very familiar with Dr. Paller and Dr. Choate, but you might not be so familiar with Denise Carter and with Doug Johnston, and they're from some of our pharma partners, and you may have talked to them out in the tables out in the hallway. But they are important parts of the drug development process, and part of the process is going to bring new treatments, hopefully, to many members of our community. Doug, why don't you go first and introduce yourself?
Yeah, so my name is Doug Johnston. I work for a company called BioCryst. It's a small biotech company that discovers and develops drugs. And my role in the company is clinical development physician. And what I do is I take the therapies to clinical trials and work with teams of people in our company and scientists and physicians and patients to design clinical trials at all stages in development.
And Denise Carter, why don't you introduce yourself?
Hi, everyone. My name's Denise Carter, and I am one of the co-founders and the chief operating officer at Choate Pharmaceuticals. I've spent my career in developing drug products based on different technologies, primarily in common diseases, and this time around, it's my first foray into rare disease development. And I have to say, personal because I founded the company, but also I've found this venture to be extremely personal only because I've got an opportunity to really get to know the community and be close to the treating physicians and the families, and really excited about what we're doing, developing products for ichthyosis. Our lead product is Netherton syndrome as well. But we're also looking at some adjacent indications.
Great, thank you. So for people in our community and their audience here who are not familiar with the drug development process, why don't we talk a little bit about what happens from basic biological research into someone forming their own company and creating a very specific therapy. Amy, do you want to start that?
Well, I think that's a great question for our drug development team here, but I would just say from the standpoint of us, it's really an important relationship because it's not just that sometimes a company will have something at hand or discover something just by chance, but often it's the opportunity to talk to physicians and scientists and understand the basic biology of disease and then be able to leverage that to develop a product. And there are many ways to try to be doing screening if you know what's happening from a basic science standpoint, or to repurpose even a drug that's already out there. And I'll stop with that. I don't know if Keith wants to add to that, but I think this should largely be, let's hear from Doug.
Talk a little bit about how long it takes from the origination of the idea all the way through to hopefully approval.
Yeah, I think the key point is it's a really long process. And I think the best way to think about it is where do you start and where do you end up? You want to start with really understanding the science behind the disease. That's the place where you can start. Where do you want to end up? You want to end up with a safe and effective therapy at the end. What's the time between those 2 points? It could be 15 years. It could be 12 years, 15 years. And the process has many steps. It starts with really understanding the science, the discovery. It then goes into designing a therapy to the very specific target you're aiming at. We want specific diseases going forward, right, that are designed for the disease itself. Then it goes into lots of laboratory tests. Before you would put it into any other studies, it goes through lots of lab tests, the therapy, to understand the properties of the drug. Then it typically will go into other types of studies, such as animal studies, before it goes into patients. And then there's a whole series of clinical trials, phase 1, 2, 3, in patients. I'll stop there.
There's been an emphasis recently by the FDA to really include patients more in the drug development process, and that's largely why you are here to meet the community. And so talk to a little bit about the importance of patients being involved in the development of the therapies and in the development of the trials.
Yeah. So from FDA's perspective, they have established the Rare Disease Innovation Hub, which allows interaction with not just the agency, but industry to be able to interact. And an important part of that is bringing the patient experience and understanding the patient journey. So as we design clinical studies, it's important that we think about the endpoints that are important for you. And so for rare diseases, as we think about endpoints, maybe in a common disease, say for itch, pruritus, FDA would typically look for maybe a 4-degree improvement in a clinical study, but they may, for a rare disease, allow maybe a 2-degree would be sufficient, because for a rare disease, it might be clinically meaningful. And it's important to know that. The only way to find that out is to interact with the community, find out what's important for you, what do you deal with on a daily basis in terms of managing your disease. And we are able to get that information by interacting with you and the treating community. Relationships with key opinion leaders are very important in terms of being able to collect that information.
You know, I should add, too, that you've had sessions with the FDA where patients have actually gone, and this just started really over the last maybe a decade ago or something, where the FDA was open to meeting with patient groups and individuals who had particularly these rare disorders, but also they do it with others that are more common. And I think that has really influenced the FDA in terms of their willingness to be flexible and to try to give some advantages when it's not so easy to recruit with large numbers.
I think one of the other points to make as well is that the FDA has changed its perspective on the inclusion of children in research. And it used to be that everything had to happen in adults and then cascade down to children. And now the FDA is looking for a plan that leads to a step-down approach that actually leads to trials in children within that same unit rather than needing to do separate trials. And this represents, I think, a sea change in the way that the FDA is thinking about disease, especially in the setting of rare disease where we have a disproportionate number of affected children and where, in particular, treatment early in life can potentially have the greatest benefit. And so I think, FIRST has been doing a series of listening sessions with the NIH and with the FDA over time, partly to reinforce these ideas, but also to represent the needs of the specific indication.
When dealing with a rare condition like the ichthyosis, it's hard to recruit people to participate. And we're going to talk about the importance of you participating in a little bit, but there have been allowances made to assist in that recruitment, things like a single arm study or basket studies. Can you talk a little bit about how that is perhaps helping the process?
Yeah, so it is hard to get numbers. We need, for a traditional study, usually, let's say we're talking about atopic dermatitis, we're talking about having 2 pivotal studies that are phase 3, so already pretty advanced, ready to go, and we're talking about, let's say 1,000 people who are exposed to this at a kind of a minimal, and usually, unfortunately, the FDA requires placebo arms for everything in phase 3 trials, a little different in Europe. And so it's really important to be able to step back and say, whoa, that's never going to happen, let's say with ichthyosis. So basket trials pull in more than one form. So let's say you might do all different forms that have involvement at the bottom of the feet because we think that there's a shared mechanism and we can do that together. There are risks to that because one may respond and not this one, and if you have a basket, sometimes you lose the ability to sort that out because you don't have the numbers that give you statistical evidence that something is working. We have done a lot of work with having each person as his or her own control. And to me, that's one of the best ways to do it. There are issues with that, but an observation period that's long enough to try to get a sense of what is the natural history in the patients that you're actually going to do the study in does to some extent allow one to then move into a phase that compares to that period of time. Now, in the future, I hope we're going to have more natural history data where we really understand each of these various subsets of ichthyosis to be able to move with that. But the only disadvantage with, for example, we just completed a trial, as I mentioned yesterday, of dupilumab, repurposing a medicine that's out there for eczema that's just incredibly safe, down to 6 months of age, and wanted to put in as many people as possible, getting them quickly on what we say the open label, where they're actually getting the medication. And so knowing that we were looking at itch and knowing, at least in the eczema trials, that that was an 8-week period before it really pretty much gets to where it's going to be, we did something a little different where we had an 8-week observation period. Then we put them in the trial, and we, instead of having a single, what we call static measurement, how are you doing at baseline? How are you doing at 16 weeks? Where, as you well know, you may not do well that day, but be otherwise doing well, or something like that. We actually took means, or it could be medians, of 2 periods to be able to do that. So I think you have to be a little bit flexible in thinking about that, but we want to, on the one hand, maximize the opportunity for everyone to get the medication as soon as possible, especially without having a period where they're flaring because we have to stop medications. I think one of the issues, I mean, there's so many things we could talk about with clinical trials, but one of the issues too is that clinical trials are done as monotherapy, which means that you're either on the medicine or you're not on the medicine, and you can't be on anything else that's going to get in the way of trying to figure out if something's working. It's not real life, because we tend to combine medicines to do the best we can to tackle a variety of different problems, especially with complex skin problems. And but that's how it's done. So it's a very awkward, unrealistic way that we do this kind of treating. So the other thing that we're trying to push through with rare diseases is the ability to stay on something that's been helpful enough that it mitigates somewhat, but you still have a severity that's enough to move into a trial. And again, we can keep talking, but go ahead, Keith.
Yeah, I'd like to address, I think, just a couple of things. So first and foremost, this is why it's absolutely critical that everyone is a member of the registry. In the past, people thought of the registry as being a way to get a genetic diagnosis, and they saw that as the primary utility. I think as we've seen this incredible investment by industry in terms of developing new drugs, the primary motivation to be a part of the registry now is both to contribute to fundamental understanding of disease states so that we can develop useful biomarkers for industry to use to measure efficacy of their drugs, but also for you to have the opportunity to be contacted when a relevant study arises. And so this is number one reason to be a part of it. The other thing that I think we will encounter as the organization matures and as interest increases is the fact that there will be multiple players within individual disease indications. And that's where having a very strong medical and scientific advisory board for the foundation is really critical, because the medical and scientific advisory board needs to use, with their best judgment, their capacity to help companies to really understand where they fall in terms of relative likelihood of success. And in fact, I don't know if Dr. Paller knows this, but we've both spoken with the same company recently, and we both said, don't do this trial because there are other things that are going to be better. And I think that that's really a service that we can play as well, because there are instances where companies can go very far and spend a tremendous amount of money on an asset that's unlikely to yield results, whereas we really need to be saving that patient population to be part of studies where we believe that there's strong scientific justification for the study to proceed and a higher likelihood of efficacy. Because one of the things that we don't want to do is have the membership of the organization engage in trials that may ultimately be futile. And this is where medical and scientific advisory boards are important for foundations, but also where medical advisors to individual companies and individual trials are also important, because they're there not only as bolsters to say, rah, rah, get this done, but also to help give a rational perspective on the likelihood that the trial will be successful, and to make sure that we're not exposing patients to therapies that will either be harmful or that are not showing likelihood of success.
You know, I'd like to add to that too, that to do good clinical trials, the worst is to have a poorly designed trial that people actually spend time in, and at the end you come out with nothing because it's not done well. And so this is part of this effort that we're working on as well. One, to have good clinical trial sites who know what they're doing around the country. And as you heard from Shed, we've hopefully established the structure, even if not funded. Boy, I sure hope we can still run with this. We've got an incredible organized structure that if there's a little bit of funding and there's interest, wouldn't it be great to not have to be asking, like, who can do this, who can do this? But to have established centers that grow over time, with shared protocols, with shared, basically logistics of how to do things. So having that is really important, a network of referring sites. Secondly, something that Keith and I have been very involved in is this concept of clinical trials readiness, and having well-validated tools that everybody uses the same one. If I can just say, for Netherton syndrome, there's something called NASA. It's never been validated. It's a Netherton Area and Severity Assessment, I guess. And it was basically just adapted from something from eczema, and it really isn't the best. And so there's been an effort to try to make that into something that would be a tool that's better, have good descriptors, do it better, but it's still a problem. And so having clinical trials readiness with good tools that really are relevant to ichthyosis is incredibly important and well on its way through some of the efforts that have been made. Rare problems, okay? We aren't just thinking about numbers. We need to think about facilitation with rare issues, because we're just not going to have those numbers. And either we've got to have funding that brings people to sites where there's the expertise to do the clinical trials, and I would say that's we need to do more with televisits. And this is another goal that we have, is to try to figure out, for example, being able to teach you how to do the photographic poses with good pictures from your phone that will help, and then have that automation that William Liu talked about. That's going to be huge. We've never had a self-assessment. How can you at home be assessing your severity in a validated way unless we have that? So those are two tools, in addition to tools where you just report out, and maybe even having a sensor that you'll have at home in a trial that actually measures your barrier function. Why are we doing these? We're doing these because then we can gather more reliable data without having people fly in to a center and really reduce costs, and yet in the end have some good tools. And so I just wanted to put out there that there are lots of things going on. Why they're going on? Because at the end of the day, we have to retain and recruit people to be in trials, and we understand the huge burden. And secondly, if we don't have meaningful data that come out, the trial is a huge investment for little gain.
I think that throughout this conversation, several times the FDA has come up. So can you talk a little about some of the fundamental shifts that are going on at the FDA in just the recent couple of years, and how that's affecting drug development and affecting our community?
Yeah, I'll speak to that. I think that FDA is more inclined to look at single-arm studies. They are also, instead of 2 duplicate studies, in some cases willing to consider a single pivotal study. So they are making allowances to accommodate the smaller patient populations in rare diseases. So that's a good transition that's happening. And I think recently they actually, just last week, announced that they're adding 2,200 staffers to the agency, which will help with the review process, which is very exciting. And I think, there's been some changes in leadership, but I feel like that, it's getting some momentum now, and I think we should think about some positive things that are happening. They just reversed one of their decisions for a company called Unicure, where originally they had presented data, and FDA said, look, we really want to see a placebo-controlled study. And after some negotiation back and forth, they reversed that decision. And they have granted approval based on an open label study. So that's all good news, and I think some exciting changes. I think with the Innovation Hub that they've put in place, it's also an opportunity for companies like Coin and BioCris to have more regular interaction with FDA, so we know what the expectation is, and hopefully can shorten the timelines, but also educate some of the investigators there. I mean, think about it. They're in the agency, so they're not the experts that are actually treating the rare diseases. So we rely heavily on bringing experts in, and they're more open to having key opinion leaders influencing what those designs should look like. And that's the appropriate approach, because who knows it better than the treating physicians, right? So there are some really good positive changes that are happening at FDA. They also reinstated the pediatric rare disease designation last year, which just supports more innovation from industry. It kind of incentivizes us to invest in additional clinical studies. So there's a lot of good things happening. You should be encouraged.
I'd like to put a counterpoint to that, and that at the same time as there are investments in the FDA, the situation at the NIH is significantly more dire. And I think it's important for the membership of this organization to be aware of the fact that using simple budgetary rules, the administration has cut back the number of new project awards that are given to investigators on a yearly basis by requiring that grants be funded up front by 5 years instead of just single years at a time. And that move seems like a simple financial one, but what it means is it's going to limit the flow of science. And remember, everything that many of the most transformative therapeutics that have ended up in industry have started in academia. And so if we are throttling the capacity to do research by using things like multi-year funding, by passing on, by reducing the indirect cost rate that goes to universities. It actually costs us about $2.50 for every dollar of research that's done. I run a department of dermatology. Dr. Paller runs a department of dermatology. A very small clinical margin is what allows us to help people to do research. We're also seeing pressure on that clinical margin by changes to the way that underinsured people are funded and or being removed from insurance rolls. It's not a moment where we're seeing a doubling down on investment in science and technology. And of course, that's the thing that has made this country great over a long period of time. And so all of you, I think, need to become activists for ichthyosis, for research. Because remember, the most basic and fundamental, and we're going to get to talking about some of the things that people want the most, like gene therapy, and there is a need for transformational technology to come about. That doesn't come necessarily from a department of dermatology. That very well could be coming from a department of biophysics. It could be coming from a department of bioengineering. And so we need to have a premise that science is, in and of itself, the most important end, and that's going to require really direct communication. And I don't think that Congress or the Senate are far off in understanding this, and yet we're not seeing either legislation or funding that's commensurate to that level of understanding, the stories that you tell about your experiences with rare disease are one of the most incredibly impactful and powerful ways to change thinking and to move hearts and hopefully to ultimately unfreeze what has been one of the darkest times for science in a very long period of time.
Yeah, let me thank Keith for saying that, because this is really the crux of the problem. And I do think that there's no question that when we're talking about Congress, for example, and beyond, that it is really the patient voice that makes a difference. When we've gone to the Hill as doctors to try to encourage Congress to put more money into research budgets, they're informed, but what really moves them is the patient voice. And I think the question that I'd bounce back to Chris then is, given that, I mean, you're well aware of what's happening right now with financing towards research. It's the word dire does capture it, and all of our grants are being cut. I was just telling Keith, one of my other grants, looking at the lipids in the barrier of skin diseases was just cut 20%. And this is what we're hearing time and time again. When I talked to you yesterday about SHED, and I said, it's got a fundable score. It's going to get approved, but we have no idea if it's going to be funded. It's because at the end of the day, difficult choices are going to be made because the budget is so low. So, Chris, can I bounce it to you to say, how is FIRST thinking about, and maybe we get some feedback here too, organizing efforts that will help to change this?
Yeah, we are, as an organization, part of the Coalition of Skin Diseases. Other organizations, large and small, much like ourselves. We just came back, you probably saw in the newsletter, we came back from our own visit on the Hill, where we visited with we had about 105 meetings across Congress and met with congressional staffers. And the big issue was NIH funding, and then some insurance issues as well. But NIH funding, and every year we go back there and we're clawing to just hold things steady, hold the budget the same year after year. So as everything's getting more expensive, the budget is slowly eroding. And then becomes the issue, we're also clawing, like every other organization. When we're up there walking around, we're walking around and meeting people in other offices that are from the cattle industry, the Eat Beef Alliance is there, and there might be other patient organizations there. There might be other professional associations, the Accountants Association. Everyone has their causes they're lobbying for or advocating for, and so it's so important for us. We were just 8 people from FIRST there, but we had to make our presence known. And then when we come back, the important part is then to have you all tell your story. I think that what Amy and Keith are saying is really, really important, in that we have to compete with these other organizations. Unfortunately, we're competing against cancer and diabetes, and we don't want to come across as like that we are competing. But it's so important for our voices to be at the same level, the same volume as some of these other organizations. So it's so important for you to, at every opportunity you have, to either write your legislator, or if they're in their local office, we really encourage you to go visit when they have local office hours, like the month of August, I believe they're out of session. So stop in, make an appointment. That's what they're there for. They want to hear from you, and they do want to hear your stories, and they're really, really powerful.
What about beyond Congress with some of the leadership at the NIH who ultimately makes decisions at the end of the day where money goes within their programs?
That is included. You heard Sean talk yesterday about the research roadmap, and that is one of the things that we're trying to get more involved in as an organization. And we talked in the board meeting this morning about how we were an organization that was pretty tiny, and we're growing, and we have the financial wherewithal now to grow, and there's some responsibilities that go along with that. And one of those is really to increase our presence, not just on in Congress, but also on the other parts of government as well, and to really enhance our relationships with people from NIH and people from FDA, and we're working on that now.
And I should just add that one opportunity that came up that you may not be aware of is that every year we have a Society for Investigative Dermatology meeting in May, and it's a meeting where all the scientists doing skin biology work come together, and many clinicians are there as well who are interested. And before that meeting, for the first time, we had a combined meeting of all the EDS, of all the epidermal differentiation disorders. And first was front and center with PC Project and one other organization that's there. And the NIH was there. There were several people from the NIH, including the acting director of NIAMS, which includes skin, who will ultimately be making the decision about how to allocate funds. So that kind of continued engagement and show and tell, so to speak, because it was an opportunity for scientists working in the area to hopefully dazzle somewhat with the need, the technology, and the output that's been happening lately over the last few years in this area. And I think they did come away from that having enthusiasm for funding this kind of work, and keep our fingers crossed.
Yeah, you have to be in the game to play the game, and so we as an organization are stepping up our efforts, and it's going to involve getting you more involved in those advocacy efforts. Bigger organizations have staff, have tens and 20s of people who work on just advocacy for those organizations, and we're not quite there yet, but we're definitely going to step up our game to get you more involved. So there's two ways we look at it when I'm trying to set up this kind of organization is the grassroots part, where you all can write a letter, you all can have Congress people, you all can let them know about your stories, and grass tops. And that's when someone out there, I'm sure there's people out there who have relationships with their Congress people on a different level. Maybe you went to college with them, maybe you play golf with them, maybe they go to the same church as you. You have a little bit extra influence, a little bit extra in to get your message heard. So we want to know about that as well. So if you have those extra special relationships with members of Congress or their staff, that's really helpful for us to know. All right, you all sent some questions in, and maybe we can move on to some of those questions before we take questions from the audience. We will have an opportunity to do that in about 10 minutes. But a lot of the questions, there were some common themes, and some of the common themes were around gene editing and gene therapy. So we had one of our sessions in Detroit, we had one of our one-day meetings in Detroit, and we were asking the question, it came up sort of like this. It would be controversial. It was like, gene therapy, hype or hope? Who wants to take that one?
Well, you know, I think that it's not just hype. I think it's hope, but I think we need to be realistic in understanding that we're not talking about something that's going to be present probably maybe even in 5 years, maybe 10 years, maybe something more. We have gene therapy moving forward for genetic skin disorders. But it's largely been in the area of a blistering problem called epidermolysis bullosa that has the advantage or disadvantage, depending on how you look at it, of having the skin barrier gone, and therefore you can get things in more easily, or if you're talking about grafting, you're getting, you don't have to take off because a lot of it's already off. And I'll explain that a little bit more so you just know what's out there for that problem and why it's different. So we have two, and I'll put in quotation, gene therapies out there right now for EB, epidermolysis bullosa, the blistering problem. And the first one that came out, not truly a gene therapy in the sense of something that's going to cure, really more taking advantage of introducing the gene to create a factory in our own skin, really a pharmacologic where once a week it's put on again. But nevertheless, because the barrier was gone to a much greater extent than we talk about barrier deficiency here, you're able to just drip onto an open area a product that has the genes, and the genes are able to incorporate not into the center of the cell, the nucleus where all the DNA to RNA activity begins, but rather just outside of that, sitting in the rest of the cell, where it's able to pump out the product and it actually gets to the right place and it can function. And it's not a miracle, but it has been helpful for many people. Then there's the other one, which is potentially curative, because what's happening in that place is that we do biopsies, we grow the skin cells to a certain extent, and it's not we who grow the skin cells, we send them off to what's a special facility that's really very carefully inspected to be able to put something back on a person. And so these cells are grown out, and then using a virus, which is the way right now any kind of gene therapy is introduced, it makes the skin cells from the patient's own skin able to express the gene that was basically either absent in terms of a product or a product that didn't function before. And now you've got a very functional product. And what we're able to get from that is up to 12 credit card sizes of skin. And when we say skin, we're not talking about skin as you think of it. We're talking about just the outer layer and 2 to 3 layers thick of that. It's not even the full 10 to 11 layers that make up our outer layer. And this comes, these very fragile sheets that come on a backing. And then when we go to the operating room, we have to actually make a rectangle that's that shape, because you can't overlap it on the normal skin. So we are taking off large amounts of skin, even a blistering disorder where it's pretty open. And I could explain it more, but I think that tells you that if we're talking about ichthyosis, with the current state of the art of what we have, we can't get it in. Or we have to basically strip your skin for each one of these that goes on, and it may or may not take. So unless you have a really high percentage of that taking, it's probably not going to be an approach. If there were certain areas, feet, for example, terrible pain, can't walk, maybe that's something to consider, selective areas. But that's the state of where we are right now. Now, there's a lot of research that's going on. We can talk about that in a minute. But I think ultimately, for something like this, we've got to figure out, for this and so many other skin problems, how to get through skin. Nanotechnology is an approach that people are thinking about very carefully, and there are some technologies out there that have not yet been applied. We're trying to get them moving forward, but where you can actually put something on the surface and enough of it gets in and gets to the nucleus that it could actually cure. Now, there are a lot of issues with what's out there right now with gene therapy, and we can move into that next, but I just wanted to give that as a preface to what is out there and why it's a problem for our community.
So I might follow by saying that when I was at Stanford, I did a lot of the foundational work that led to the therapeutics for EB. And EB is a unique state. And so are most of the diseases that are being treated right now using gene therapy. And that is, they exist in cells that have the capacity to expand within their environment and to replace the diseased cells. So until we can master this art of creating competition between corrected cells and affected skin, it's going to be hard for us to do treatments using current technologies for ichthyosis. The other possibility, and I think that this is the other thing that Dr. Paller is really driving for, is we're seeing myriad people who say, oh, we can get things into the skin when we do it in skin equivalents in culture. And yet, when they go to the epidermis of real individuals, they can't get their product in. And so we need to have a high bar, we need to have skepticism, but we also need to have a lot of enthusiasm and support for the people who are developing these transformational technologies, and remember the fact that it's really supporting the most basic research that ultimately leads to the translational discoveries. I'll also point out the fact that many people in this room have some of the most common genetic diseases that exist in the world: high blood pressure and hypertension. We don't treat either one of those with gene therapy because we have highly efficacious molecular therapeutics to treat those diseases. This is why we are working so hard to advance our understanding of the fundamental biology of ichthyosis and using these remarkable new technologies that we have in the form of proteomics, microbiome analyses, lipidomics, spatial transcriptomics, single-cell biology to understand how individual gene mutations lead to specific defects in cells over the course of making normal skin, and also seeking to discover how there are similarities across the different genes that cause ichthyosis. Because I think that the goal is ultimately to develop therapeutics that are not bespoke to specific mutations, but to develop therapeutics that are going to lift all boats simultaneously. And I think that's one of the reasons that we are so excited about having all of you be a part of the research that's happening at this meeting to the degree that you can. Because it's not just about supporting the registry. It's not just about supporting the work that's happening in SHED, in my laboratory, or in Dr. Paller's laboratory, but it's the network of investigators that access this data and access these resources. What we're trying to do is create a foundation for research that's going to facilitate the development of the next generation of cures. We have been able to, in specific cases, repurpose drugs for the intention of treating specific diseases. And we've seen this has been particularly effective in a couple of types of ichthyosis right now. And I also believe that having this comprehensive data set allows us to very rapidly translate what we find from spatial transcriptomics to looking at drug databases and to be able to predict in real time what are going to be efficacious therapeutics. And then beyond that, the other side of it is trying to figure out why is it that some people respond to certain drugs that are repurposed and other people don't, and understanding what those biomarkers are. And so we talked about a decade of discovery, and I'm looking into the past. I actually think that the most important decade is the decade that sits right in front of us. And in fact, probably just the 3 years that are in front of us. And so, while we may be saying gene therapy at this moment is not the thing that's going to be the cure, there may be a discovery next week that totally flips that script. And so, the message that we have for you, I think, is one of engagement, one of hope, and one of promise, because we are seeing a capacity to do things that we haven't seen before. One of the questions that was in the chat was whether AI is going to be able to do this. The problem with AI is it's very good at summarizing things, but it's not very good at creativity. And it's still all these young people that you see who are here at the meeting, who are out in the halls. It's about taking scientists who study completely different things and getting them excited about ichthyosis. It's conversation. It's these sort of unexpected serendipities that happen in the world of science that are really the driver. And I think that the organization has done an incredible job of focusing on pilot grants that bring new investigators into the program, investing in new technologies, and in investing in individuals who have the potential to change the field.
That's great. Can I just follow on that with I just want to explain what's so incredibly special about this conference, and thank everybody again who's done anything with research, whether that's anything from biopsy to non-invasive to surveys. We really need this. This is unique, and this is going to give us an edge. Now, when Keith talks about all these multi-omics, which are so great that we can take tiny pieces of skin or little bits of blood or whatever it is that we're doing and get huge amounts of information, it's also having the clinical information that's so important. So for example, we're looking at omics analysis, but because we have information about transepidermal water loss telling us about the barrier, because we know how much you're itching, because we know how red your skin is, because we know how much scaling you have, each one of these parameters as examples can be correlated with data that we're seeing that's up, that's down. And so we can start to look at subsets. We can also start to look at clinical data that correlates with changes that we're seeing and start to understand how we can use something more for the redness or something more for the itch or that sort of thing, and really do it scientifically. So I wanted to just mention that because what we have that's special is we're getting things here, but we are also getting your feedback on how red you are. We may not be examining everybody here at this meeting. Maybe that's something in the future, but it's just, we can get so much data from you that can be correlated then with small samples. So I wanted to mention that. I want to say one other thing about repurposing, just because you've heard that word, and understand why we're trying to do this. There are two ways to move forward with having medications. One is drug discovery. So a lot of what we're talking about here of looking at pathways, what do we need to find, or just basically taking cells with robotic methods to just have large number of culture wells and adding all kinds of different drugs that might be possible out there. These are all techniques that we're using right now to look at drug discovery, and ichthyosis should certainly be part of that to a greater degree. But repurposing is something that's based on the pathways that are discovered, and we mentioned some of them yesterday. You keep hearing about these. There are drugs already out there. There are drugs that are being used for other skin disorders. There are drugs that are being used for oncology, for example, and a range of other fields that we could potentially repurpose. And if we're repurposing them, what does that mean? Why is that low-hanging fruit? Because number one, it's already gone through testing. It's gone through the testing of cells. It's gone through the testing of animals. It's gone through the testing of people. So if I can think of a medication that's out there that might be useful for this community, I can go to the FDA with what's called and get what's called an IND in 30 days. They don't even question it. If I want to try to do a study and repurpose it for an age group, for a demographic it's already approved for because those safety studies were done. Versus how many years to get to that point can be a decade and millions of dollars of investment to get there. So in the end of time, it's not going to be the only direction. But this is why we keep thinking about pathways and using drugs that we can repurpose, whether that can potentially be small enough to be put into something topically delivered, or whether it's delivered systemically. And sometimes we'll even take things that are out there being given orally or injected and turn them into something topical, because who can use that but us in skin, right? So that's just, I just wanted to explain those two things a little bit more.
Yeah, Keith, you used the word investment in your most recent remarks. I think there's a lot of a feeling within the community that because we're rare, because there's fewer people, it becomes down to a money issue. There's not as many resources, and we know that money can't make you happy. Money can't buy me love. Can money buy us a cure? If you had more investments, or if the NIH gave you, tripled your budget, how much of a change would it make? And what are the non-monetary barriers that are slowing the process?
Well, non-monetary barriers, we talked about, getting people. Clinical trials are everything's expensive, but that's a magnitude of greater expense when we're talking about doing a clinical trial. I'm not sure people realize how expensive, and maybe we could hear a little bit about the cost of clinical trials, but that's where it starts to get even more expensive. So all this preclinical work has a certain amount of expense, but the clinical work, where's the money for that? And you'll notice the companies are not necessarily huge companies. Sometimes we're talking about startups, very focused perhaps on only ichthyosis. Where do they get the money to do these studies that some giant company out there that's rolling in money could make an investment more easily, right? So I'd actually love to hear a little bit about that.
Well, companies usually get investments different ways, right? So I think in a small startup company, there might be a reach out to investors to invest in the company, and that helps them then get the resources they need to do clinical trials. Other companies, we have at BioCryst, we have a therapy that's on the market, so income being generated from the sales of that product is helping to fund our clinical development program in a variety of rare diseases that we're working on. So I think it just depends on where the company and the stages that they're in.
What's the cost for a clinical trial that is at a level that it could lead to something becoming commercially available?
So a phase 3 pivotal trial that would generate data that we could take to the FDA and get approval of a drug is a multi-million-dollar venture. And depending on
Tens of millions.
yeah,
For a rare disease, tens of millions.
for a rare disease, it's still tens of millions of
Even though there are far fewer patients that are required.
We get calls from companies like BioCryst or Coin asking for us to help with marketing the recruitment of trial participants, and they have these timelines. They say we have 12 months to recruit 70 people, for something like that, and we say 70 people. We should be able to do that in a couple of weeks, and it takes 12 months. And sometimes we fall short, and so that's another message out to you that when a trial gets presented to you, and it's on on our website or it's on clinicaltrials.gov, really take a good look at it and ask questions of us or ask questions of the company about how you might participate, what the interaction might be. We'd love to be able to answer those questions, and because the longer it takes to fill that trial, that's dollars as well.
Can I just ask a question? So you depend on investors a lot, and investors talk to us, and that's great. But would there be value? Would there be a panel of people available to talk to investors, including in a Zoom meeting, for example, where they could really see what's happening? Each person here has a story, and those stories are very valuable. And I don't know how much investors have access to that, but I'll bet that would sway their decisions.
I can answer that because we've actually started a Netherton Now campaign because a lot of investors weren't aware of the disease that we were focused on developing a product for. So we have started to tell individual stories. And we have started this awareness campaign. We started it February of last year. Our first video went live on Rare Disease Day. We have 6 videos out there now. We've just done another round of filming. We will do another round at the end of the summer, but we have to continue to tell the stories. But we did a round of fundraising last year, and I can tell you that we were talking to a number of investors, and we started to talk to them about the awareness campaign and how important it was, and they said, we don't even need to know. That's how we found you. So we are actually out there telling these stories, and it is important that they do hear your voices. And so if there is an opportunity to do a panel or things like that, it's very, very powerful, and should definitely, if somebody's reaching out to you to participate in that, please consider it.
But of course, at the end of the day, this is very much about money, right? And so if you look at one of the most successful recent raises, it was by Verderamics in New Haven, which is essentially making a timed-release version of minoxidil, which is $1 a day drug, not going to change the world, and yet they raised $768 million. And so there is a disconnect with the way that we ultimately are supporting and funding research with the idea that this is it's a for-profit enterprise, and that we are using sort of a capitalist approach to the development of therapeutics for rare disease. And it raises the question of whether we are thinking clearly from a national perspective on how it is that we fund rare disease research for disorders for which there isn't going to be a whole population that's going to ultimately take the drug. So, food for thought.
Now it's our turn to have some questions from you. We have a few more minutes left. If you want to, there's some microphones right there. Jacqueline, and if you could say who you are, that would be helpful.
Hi.
Just to give you one, we can't really see you very well.
Okay. I'm Jackie Katsoukas, and my quick question is, so going to Congress, can we have a sample letter so that we know what to say? Of
Absolutely,
course,
yes.
we'll rewrite it.
Yeah, I think there might be some on the website already. I don't know if in
Okay.
the new website refresh if they made the transition or not, but we have had information like that. We absolutely can get that to you.
Okay, good. Yeah, because I wouldn't know what to say, and I want to do it right.
You know, one thing to note, though, is it's interesting because, of course, you get something, it's very easy if you can just kind of sign your name to something and not personalize it, but feel free to change things and personalize them. I've recently heard of something where they only count each different letter. And if it's the same letter, they count it as one.
There's
so,
all kinds
so
of
in this day and age of ChatGPT and other ways to easily change things, please do.
Yeah, there's a lot of automation going on that. So you just type in your zip code and it'll send a letter out. So absolutely, if you customize it to your personal story, it's way more impactful. Any other questions out there?
Hello, I'm Steve Mike. This is my first time here. We've heard a lot about money and fundraising and trying to contact congressmen and others, and part of the biggest issue is having such a small percentage of people and convincing big pharma or bigger companies who make a profit to try to put research into this expensive endeavor. We've also talked about repurposing drugs from other issues, like skin issues, of eczema and things of that sort. Is there any way to inversely market or pitch or convince people? Is any of this research can be inversely used to help other skin issues or the skincare industry, such as I'm just thinking kind of off the top of my head is, a lot of us have issues with blistering and healing, and can this research help with wound care in the future, or and pitch it to those companies? Hey, we have something that could help heal a simple cut 10 times faster, or we have some skin issues that are way under the dermis from a biological standpoint, where you see commercials all the time of as we get older we're getting more as a general public getting wrinkles and the loss of collagen and retinol, and pitching it as, hey, some of us have family members who are producing skin at a ridiculous amount of rate, where half the world would love to have this issue. when you pitch it like that, hey, we have something that can help isolate, reduce growing this dermis fast, and wait, we figured out the trigger, the mechanism. Would you like to be involved in this? And does that make sense?
Yep.
So perfectly stated. I mean, I think that you have hit on the fact that the biology that we're dealing with monogenic single gene diseases like ichthyosis is relevant to common disease and to skin health. And that is an argument that scientists have been making for decades. And I think that it's also one of the primary reasons that many companies have entered into the rare disease space, because they recognize that they can springboard from creating a rare disease drug that can ultimately be used for more common diseases. And we want to encourage that. We want there to be a secondary motive that allows them to see a larger market. And so I think that you've hit on one of the primary messages that we have, I think, as a community reinforced over a long period of time, and has been a driver of investment. And every grant that I write about rare disease, it's always about revealing fundamental mechanisms of skin biology that are going to be relevant to all Americans and all disease.
And I do think we need to think about this as we move forward. I'll give you an example of outside of this field. So there are tumor syndromes, for example, that involve the hair follicle. Hair is very big right now. This is why there's this huge investment in this minoxidil product. But I know a scientist who was able to separate out the fact that he could get hair to grow from the malignancy, right? So that's golden, right? Because then you can use a product that makes hair grow in scars and not have to worry about malignancy. This kind of thing too, if we can, how do you make skin heal faster? Boy, we know that you got to get those stitches out sooner. The skin's growing so fast, right? If we could harness that in such a way that we could put a brake on it so it was just the right amount of increase, then that would be amazing for wound healing, right? So that's just one example, but that's how we think. I mean, we have to think bidirectionally.
Samuel, you had a question?
Thank you, everyone on the panel. My name is Samuel Simmons. Speaking of the nanotechnology that was mentioned, there's some work going on in Germany that's in the preclinical stage. And I guess two questions, if I could. One is I think that work that I mentioned in Germany, the nanotechnology, I think that you're talking about, is in the preclinical stage and is up against a financial barrier to get into the clinic, right? So what are some ways that drugs in the preclinical stage overcome, the financial barrier or obstacle to get into the clinic? And then a question that was posed earlier, I think you had asked this, Chris, if what are some of the, in clinical trials, what are some of the non-financial barriers, obstacles that have to overcome in clinical trials? I don't recall if I heard an answer to that, so two questions there. So,
I'll take the nano because I've been doing nano for years. And I think that there are 2 points to make about how to move that forward. One is that there are a lot of people out there who have great ideas. They publish in non-medical journals, and then they move on to the next one. And I think we don't know enough about who's out there and who's doing interesting things. I mean, personally, I worked for a decade with the head of, we have a very strong nanotechnology program, and I worked with the head of that for years, and he has a product that goes through skin, but never really, and I did those studies in my lab, so I can tell you that. But never really got to a point where we were working with anything involving ichthyosis or anything that moved forward. And we've been revisiting that now because there's so much interest now in moving that forward. So there's this big gap with all of these nanotechnology engineers, getting them together with clinicians and actually getting to a point where it's anywhere near the pathway towards clinical research. So that's one problem. Two, a lot of these in the nano world are creating spin-off companies. I mean, they're creating their own small companies and getting good investment. Three, NIH funds are hard to get, but they do like looking at new products like this, and there are some specific mechanisms where you can team with these small companies in academics and try to move that forward, and hopefully move that forward enough that it gets to a point where then a company is interested in going the next distance. Because you have to get to a certain point where you're actually showing some preliminary data that's strong enough for a company that can get it through the clinical research stages go. So there are multiple steps, but we need to focus on finding people doing the good science and connecting them with the clinicians to get it towards ichthyosis, helping them with investors in the small spin-offs that they're making, getting to the point where you have enough data to excite somebody who is then in the position of the folks up on this panel and moving it forward. I think there's good stuff out there. We just have to find those connections. We should be having panels with people who are doing nanotechnology research, people doing engineering research who can help us in other ways. But I think it's out there, Sam.
But we also need to be honest about our science. I think that we are, in research, often sort of in this environment where we need to really focus on the positive and not talk about the other side of that. And the fact that the study hasn't moved forward may tell you something actually about the study itself, rather than it may tell you something about the study itself. And so, because we are in this environment of tight funding, I think that many people are taking to LinkedIn. They're really like trying to use a social media approach to fund themselves. And I think one of the challenges that we have is, as a patient organization, how do we translate the incredible promise of these technologies and frame it in the reality of where we are biologically today. And so the idea is to support transformational technologies, to create the connectivity, but then also to have a strong MSAB that serves as a translator of the, I don't want to call it the hype, but of the enthusiasm back to the world that these guys are in, trying to actually bring products to market.
And I think our experience has been, and I know Keith and I have both experienced this, we spend a lot of time talking to people who are non-biologists, and there is a gap, a huge gap, and we learn from them, and they learn from us, and so many things that come out are totally unrealistic. And these people who are inventing nanotechnology products or engineering or bioengineering products don't have any concept of what is realistic and what are problems. So, for example, we want, if we deliver something topically, we need to get it where it needs to go, and we may not want to get it in certain places. How do we control for that? And that becomes particularly important, too, if anybody's, there's just getting it into the cell and making something, that concept, or blocking something. And that's easier than actually changing something permanently. But there are some technologies, if you can have targeting sequences that get something into a nucleus. I worry a little bit more about control and getting things in the right places and making sure to be able to turn something off if we need to do that. Some of these things, too, that's technically more challenging.
Very quick example. I was asked to review a paper about a nanotechnology foam that delivered to the skin. And it showed great results. But they had to do dermabrasion on mice. And I said, well, show me the images of that dermabrasion. And what happened is they were completely removing the skin of the mice to make it work. And I said, and this was submitted to a major journal. And I said, well, show me that you can apply it just to the skin of the mice and get any efficacy. And they had no efficacy, right? And so that's the problem. We're very good at showing the very best, and it takes someone who knows the biology to be able to translate that.
Yeah, good example.
We are out of time. I'm sorry, everybody.
Wow.
We
And
could
I was
talk
just,
forever,
I was just going
right?
to add to that mouse skin is a few layers thick. Human skin is not, and you have to do the studies in at least human explants as a start to even know if it could possibly get through human skin.
I want to thank our panelists. We're out of time. Sorry we didn't get to all the questions, but I want to thank our panelists, Doug, Keith, Amy, and Denise. Very, very informative and very, very helpful.